be talking about computational genomics and comprehensive genomics in clinical applications. So we all know cancer classification has undergone a paradigm shift already, and we have moved on from organ-based classification to histopathological to the era of molecular classification today. And in this molecular classification, we want to identify the therapeutic alterations which are driving the tumors, and these drivers are further targeted by small molecule inhibitors specifically designed for those alterations. Furthermore, we have seen that several different therapeutic targets have been reported all across the world, and all of these targets are to be incorporated in a clinical practice. So how it usually goes is that a patient sample is received in the clinics, which goes for targeted NGS testing, and this NGS data is to be analyzed by computational tools, and that generates a report of therapeutically relevant alterations, and this report goes to molecular tumor board. In this report, what we are looking for is to classify different kind of point mutations, indels, copy number alterations, gene expression, fusions, as well as pathogens, to be classified into whether it is having a non-clinical significance and association, or whether it is falling into unknown clinical significance, or VUS. For all of the variants of non-clinical significance, we further look at whether FDA and other global body approvals are in place, or whether there is any other prognostic significance, and so and so. Now, to do these, sorry, what we have to do is either go for sequential NGS testing. As you can see here, for lung cancer, we have been doing for quite some time now. Since day one, we go for EGFR testing, and then after a week or two, we further go for ALK, ROS1, REST, and if all of them are negative, then we go for MET, and so and so. So this usually takes about 30 to 40 days for analysis of 7-8 different biomarkers in lung cancer. Instead of that, if we go for comprehensive genomic profiling from day one in the patient, then within a week we get to know what is the mutational landscape, and that saves a lot of time as well as tissue. Now, coming to the tissue, how that is actually helping. So each of the individual assay that we perform in the laboratory requires roughly about 3 to 8 different sections of FFP block, and combined together, this entire non-comprehensive genomic assay will take roughly about 25 to 30 sections minimum of a typical patient. If you go for comprehensive genomic sequencing right away, then we can make the same comprehensive information available at the expense of about 5 to 7 sections only. So that saves a lot of tissue, and this can be very critical for cases like lung cancer, where getting a large chunk of tissue is a major problem. Apart from that, we also have to make a conscious decision of which genomic test we want to use, and there are several options available. Here I'm showing you a very small list of the more widely used test available, which includes Foundation 1, MSK, Caris molecular test, and Illumina TrueSites. They all range in the order of 300 to 500 genes, and recently our Indian company Care has launched the 1200 gene panel. Whichever genes we go for, all of these are considered to be comprehensive gene panels, and how these comprehensive gene panels can cover the alterations which are not otherwise picked up. So here I'm highlighting a few of the studies which are showing the total yield of therapeutic targets, and you can see that in this latest study from 2022, which was from Japan, around 23% patients of various cancer types were attributable to targetable therapies. Another study from India, having 297 patients, was showing about 10% of the patients receiving targeted therapy. This number is slightly lower in this entire table, because we know that we have certain resource limitations in the country. But if we look at other countries, mostly data from the USA, we get to see that roughly about 35% to 70% of the patients are attributable to targeted therapeutics. But these numbers can be very tricky, and these numbers can be very tricky because getting this information is also a big challenge. So here at Tata Memorial, when I was working with Dr. Amit Dutt as a postdoctoral fellow, I developed a tool called CLINO for clinical genome analysis. In this CLINO, the first question we asked was across the whole genome, what are the therapeutically relevant biomarkers and how we can catalog all of them in a single comprehensive NGS assay. So we have at present about 175 genes, which covers roughly about 3,000 different mutations. And all of these mutations correspond to 283 different drug compounds, and there are about 26,000 gene to drug relationships in this database. So by building this database, we are now able to catalog what is there actionable, what is available, and how we can take advantage of it. Furthermore, we implement this into various kind of situations. We simulate how this therapeutic report can be interpreted. So, say for example, in this case of lung cancer, if there is a EGFR L858R and T790M mutation detected, then CLINO will recommend osimertinib, and it will also highlight all the drugs which are already known to show resistance because of presence of T790M. In a secondary case, where a lung cancer patient harboring FGFR3 alteration, CLINOM is able to recommend a drug called BGG398, which is under clinical trial, and CLINOM is also highlighting and giving a URL. One can go to the PDF, click on the link, and they will be presented with the whole set of clinical trial details, and that can be recommended to the patient if it is feasible. In this third case, it is an endometrial carcinoma patient harboring EGFR L858R mutation, which is not a conventionally expected mutation in endometrial carcinoma. In such case, CLINO will recommend you to use gepitinib and all other EGFR TKI. In the off-label setting, it will also tell you that the approval for these drugs and for this mutation are for lung cancer and not for endometrial carcinoma. So this is not a standard practice, but this can be really useful in some rare and challenging cases where no standard options are available. And last but not the least, cancer pathogens have also been shown to have therapeutic implications. Most notably, here I'm showing oral cancer patient harboring HPV infection, which is recommended to undergo aggressively reduced radiotherapy. Otherwise, they have much higher toxicities and all. So that is a level of personalization we can provide even to the non-biomarker driven therapeutic decision making. Now, to make all of these different kind of reports, we have to use a computational algorithm. And while developing this algorithm, we realized that there are several challenges that we practically face in decision making. So how that happens is that we get the NGS sequencing data, which goes through the standard practice, best practice of genomic data analysis, and we identify biomarkers from there. Now, after identifying biomarkers, we need to ask three different questions as shown in these green boxes. And to ask these questions properly, we need to know the cancer type of the patient. Now, say for example, pathologist one would like to call a cancer adenocarcinoma of lung, and pathologist two would like to simply write lung adenocarcinoma. These two texts means same to us, but it doesn't mean the same to computer. When you try to make these two text strings, it will not be identified as the same text string by the computer. So what we do is we use a natural language processing algorithm here, which is a AI algorithm. It takes a different kind of text strings, normalize them, and interpret them, and the interpretation is then matched together. So that helps us identify the right cancer type, irrespective of what pathologist is reporting in their pathological report. So after identifying that proper cancer type, we ask whether biomarker is already known for the same cancer type, whether biomarker is known for any other cancer type for off-label utilization, and whether biomarker is part of any ongoing clinical trial. Now, at this stage, again, we have to consider the cancer type recommendation from the label recommendation, whether it is coming from FDA or any other organization, there also again the text strings are mentioned in a different manner. So for example, EGFR, erlotinib and gepitinib recommendations are always identified as non-small cell carcinoma positive patients. And nowadays, we don't use non-small cell carcinoma terminology. We directly classify a lung cancer patient as adenocarcinoma or squamous cell carcinoma or small cell directly. So this adenocarcinoma terminology will not be found on the label. So how will you match a label with your cancer type, which is coming from your pathologist? So we developed another AI tool, which is completely in-house designed at Tata Memorial. We call it tumor type tree or T-cube to call it short, where we phylogenetically in a child-parent relationship wise, we link 800 different cancer types, through which our AI algorithm is able to understand that any label approval for non-small cell carcinoma will be equally applied to all the child terminologies within non-small cell carcinoma. And because of that relationship awareness at the computational algorithm level, CLINO is able to recommend EGFR TKI irrespective of what child classification of lung cancer you are recommending, irrespective of level complexities and so on. So this is able to sorry, this is able to get rid of these anomalies that we get to see when we are trying to match label with the current practice in the field. Moreover, it is also able to take care of tumor agnostic and tissue agnostic drug recommendations. And to our knowledge, this is at present the only tool which is able to do so, which is available open source. Say for example, NTRK is recommended for all the solid tumors. Now, when we say all the solid tumors, as a human being, we know what all comes under the umbrella of solid tumors. A computer doesn't know the same thing. So by using this tumor type tree relationship, our CLINO is able to understand all the cancer types irrespective of what level of approval is available for them. So those are the selling features. Now, apart from that, we have developed a GUI, a graphical user interface. So a user doesn't need to go through command line operation of the tool. They can just select appropriate options, click on the OK, and as shown in this video, within few seconds you will get a complete comprehensive clinical report as a HTML file. We have also implemented few computational engineering concepts to make it faster and compatible for smaller computer systems, so that it doesn't depend on very heavy high-end computational systems all the time. Although having a better computers will be always helpful and recommended. To validate the findings of the CLINO, we have analyzed 11,000 patient samples from TCGA. And what we have observed is that roughly about 10% on an average are attributable to approved therapeutics, as can be seen here. And in few of the cancer types, we can see that this percentage goes very high as 70%. When we consider the approved and off-label therapeutic considerations, this percentage increases to about 18%. And when we consider ongoing clinical trials all across the globe for precision medicine, roughly about 40% of the patients are directly attributable to one or another therapeutic option. So that gives us tremendous hope in the near future. So that brings me to the final slide. We have seen how comprehensive NGS analysis right away can save a lot of time and tissue for the patients, and how it can help us get a more comprehensive idea of therapeutic options. And we have also seen that how pairing this comprehensive sequencing with CLINO type of computational solution can help us get the broader picture and more efficient identification of therapeutics across all the cancer types. Thank you all.
Thank you, Dr. Pratik, for giving an understanding about the CLINO which we have developed. Any questions? Okay. If not, thank you, Dr. Pratik.